Hello, my name is Alan Evans. I'm a professor of neurology and psychiatry at McGill University and the Montreal Neurological Institute. It's a pleasure to give a keynote lecture at the World Congress of Psychophysiology in 2021. I'll be happy to take questions in the live Q&A session. So I will be talking about open neuroscience. I come from the McConnell Brain Imaging Center at the Montreal Neurological Institute. It's been around for 35 years now. And my talk outline will be as follows. A brief introduction. I will talk a little bit about a large program that we have at the Montreal Neurological Institute, McGill University, called Healthy Brains, Healthy Lives. I will then go on and talk something about the basic neuroinformatics infrastructure that is underpinning this large program. And then I'll talk about other applications in the open science arena which will take advantage of this infrastructure, and then a short conclusion. So to start off, we're now living in a world where we are surrounded by data of all scales, from subcellular up to whole brain and everything in between. We also are living in a world where there are lots of various types of data. I'm showing here some examples of imaging data available in the open science arena. Of course, there are many other types of open science data, genetics in particular, but I will be focusing mostly on the imaging context. So we have all of this scales of data, different types of data. Open data is available, and we're swamped with data overload. So we have to put in place the means to allow us to manage all of this data. We also want to respect the principles of FAIR, otherwise known as findable, accessible, interoperable, and reusable data, so that data that's placed in the public domain is useful and available to a wide community of scientists, so that science and society can get more bang for their buck by sharing the data widely than just a few limited projects and investigators having access to the data. The Healthy Brains, Healthy Lives initiative at McGill is it's a large program, $84 million from the Canada First Research Excellence Fund, and it uses big data principles, neuroinformatics, and computational modeling to address questions in neurodegenerative disorders, cognitive neuroscience of brain plasticity, and population neuroscience and brain health. And this largely refers to psychiatric and social science applications. Now, as you can see, there are over 100 investigators involved and lots of core facilities. And all of this has to be supported to provide us with the overall goals that we are undertaking to transform many brain disorders from terminal or lifelong affliction to treatable or even curable conditions, to reduce the human and socioeconomic burden of brain illnesses, and improve mental health and quality of life and productivity of people around the world. The overall goals that we have here are to understand the individual brain using multimodal information, genetic, epigenetic, physiological, imaging, behavioral, environmental, clinical, and social science data. And so NeuroHub is the analytic platform that we've developed to support this infrastructure, to support these many hundreds of scientists, trainees of those scientists, and the wider application environment. There are also goals for clinical translation, commercialization, and by 2022, a Canadian framework for brain health. The neuroinformatics and modeling component is core to the three domains that I just talked about: neurodegeneration, plasticity, and population neuroscience and brain health. And the infrastructure that we've built to support this NeuroHub is illustrated here. And what you see, notably the area over here, the Loris Seabrain Datalad area, this refers to the platforms that make up NeuroHub. Of course, we also use NeuroHub to support data dissemination, data exploration, open publishing, and big data processing. It's not just about data storage and dissemination. You also have to provide the means to analyze those data in place. So just to reiterate, I'll be talking for the next little while about the LORIS databasing system on the left-hand side of the screen and its partner, the CBrain high-performance computing platform. Now, LORIS, as you can see here, supports a range of different data modalities, including all forms of brain imaging, genetics and epigenetics data, and I stress it's really the summary data, the SNPs and CNVs, not the full raw data of genetics and epigenetics, that are purpose-built genetics databases which can store all of the raw data. What we are concerned with is data that is usable within the context of a multimodal imaging and multimodal analysis environment, including imaging, behavior, genetics, and so on. So we have the ability to store a lot of SNPs and CNVs, but not all the raw data. Behavioral and clinical information, psychometric testing, psychological, psychiatric tests of all forms, biospecimen information, I'll come on to that as we speak later, and electrophysiological information. So all of this data can be stored and queried and made available for data dissemination or downstream processing. And in that regard, we are using, I'll talk about CBrain, which is an orchestration portal which allows us to get access to high-performance computing platforms and indeed cloud services. And of course, we can then do the open publishing using techniques like Jupyter Notebooks. So LORIS is one of 3 or 4 systems with a global footprint. It supports many national networks. CCNA is the Canadian Consortium on Neurodegeneration in Aging in Canada. I'll talk about the Canadian Open Neuroscience Platform, CONP. IBIS is the Infant Brain Imaging Study. ABCD is the Adolescent Brain Cognitive Development Study. These are all large programs. Multi-domain imaging data storage, I've already talked about. A user-friendly data querying tool. It's now reached over 200 man-years of development since 2000. We typically have a team of 10 to 20 software developers who are working on the continued evolution of LORIS and CBrain. So LORIS is interface with CBrain, as I will talk about later. And both platforms are open source and published extensively. You see on the right that the various features that LORIS offers: study and behavioral data management, genomic biosamples, laboratory information management systems for biospecimen information, queries and processing, imaging data management, standards management, and notably electrophysiology. LORIS is an acquisition management environment as well as a repository for data download, so it can be used to manage multi-site projects, either in the academic domain or the commercial domain for clinical trials. And of course, as I've just been talking about, it's a front end to various analysis pipelines. We use it for CBrain, but there's nothing limiting it in terms of its ability to interface with other analysis environments. LORIS is widely used in many projects around the world, and at this point it has over 1,000 different behavioral instruments already coded and an instrument builder which allows us to add in new instruments for any new project. CBrain is a high-performance computing portal, and its general principles are that data can be anywhere, computing can be anywhere, and users can be anywhere in a distributed architecture. You see on the right a schema which describes the user at one end and the compute resources, be they cloud or big iron at one end, and the data servers. All of this information is handled through the CBrain portal layer, which essentially removes a lot of the heterogeneities of individual platforms out there, different environments for high-performance computing, and makes it relatively straightforward for a not necessarily tech-savvy neuroscience user at this end. Boutiques is an environment that was developed by a colleague at McGill, Tristan Glatat, and this allows us to containerize quite complex workflows in a relatively straightforward manner into Singularity or Docker containers for wide distribution across the individual HPC installations. The CBrain network was originally set up in Canada to bind together the high-performance computing platforms in the country. And at this point, it's about 400,000 CPUs across the country, which by Canadian standards is quite large. By US standards, it's not so large. And we have nodes to a number of US high-performance computing sites like the Texas Advanced Computing Center, the Pittsburgh Supercomputing Center, the San Diego Supercomputing Center, and now the Minnesota Supercomputing Center. These are all accessible through the CBrain portal for scientists working in the US context. We also have nodes, CBrain nodes, with the European eBrains equivalent of CBrain, and for instance, Surfsera, which is a HPC site in the Netherlands. So it's quite generic as a HPC portal. I just talked about boutiques, and so just to emphasize the point that boutiques is a fully automated environment for deploying heterogeneous computing resources through containers. At this point, there are over 100 different tools which can be accessed through the CBrain portal, and all of these will run on whichever HPC sites are selected either by the user or automatically by the CBrain orchestration portal on your behalf. An example of this is shown with our colleagues in Minnesota, who we're working with regard to the ABCD study that I mentioned. And what you see here is emphasizes the point that the data doesn't leave the country. The data may be stored in Minnesota, as you see here. A push list is sent to CBrain, but the data is transferred, for instance, to the Pittsburgh Supercomputing Center in the US. None of the data leaves the country. Only a push list leaves the country and comes to us in Montreal to coordinate this data transfer both into the supercomputing environment and then back out again. The environment that I've been describing also has incorporated Datalad, which in essence is a Python tool that builds on top of GitNX and extends it with an intuitive command line interface to enable transparently operating and managing of data sets. And if the data is in Datalad, it is now automatically available for processing within CBrain. CBrain is widely used at this point, over 1,200 users in about 200 sites from about 30 countries, and steady growth, as you can see at the bottom here, in the user base of CBrain. Just tangentially, CBrain has nothing to do with the brain as such. It's simply an environment for processing large amounts of data through a distributed computing network. So it can be used for many different applications beyond neurological applications, as shown by some of these examples in finance, meteorology, cosmology, traffic control, aeronautics, and so on and so forth. You can register to become a CBrain user relatively easily. Go to this site to join in. There are a lot of publications to describe the environment that I've just described. The CBrain, Loris platforms are all open science, and openly available source codes can be accessed through GitHub. So you can go to any of these papers. You can go to the NeuroHub references list here to look up these papers and find out more technical details about the Loris CBrain environments. I'll now move on to the use of these platforms in a variety of different projects. I've already talked to you about Healthy Brains for Healthy Lives, HBHL, and I'll talk about a number of different applications that take advantage of this environment, as shown here. For instance, the UK Biobank, which is obviously a huge resource from the UK, and they had originally made this available on a per project basis to people asking specific questions. And as you can see here, the UK Biobank allows, it can answer many questions. You have data from over 40,000 subjects for MRI, and genomics in 500,000 subjects, and a lot of demographic information and health-related measures. And this is a huge resource. But it's only in the past been made available on a one-by-one basis. In the HBHL context, we made it available to the entire community. So this UK Biobank is stored within the NeuroHub environment. Also within the NeuroHub environment is the ABCD data is now available. ABCD, the Adolescent Brain Cognitive Development Database, is a US initiative. It has over 10,000 subjects, at this point over 200,000 image files, and almost 300 different clinical and behavioral questionnaires, and 10,000 executive MRI summaries, and 73,000 variables. So this is a huge resource. This will be made available through the ABCD network in the coming years. Within our own context at the Montreal Neurological Institute, we have the Tanenbaum Open Science Institute. This is a new way of doing science, and the whole idea here is to make not just a data set open, but an entire institute. And this initiative is being led by Guy Rouleau, shown on the right here, with Larry Tanenbaum, a major donor, and our present Prime Minister, Tristan Trudeau, and this is Suzanne Fortier, the president of McGill University. So this is a major initiative in open science, and one of the principal platforms that support the open science initiative is what's known as CBIG. This is the open science databasing infrastructure for TOSI. And the CBIG repository is led by Jason Karamchandani at the MNI, and this is the biospecimen repository. So this is distinct from the imaging, behavior, genetics, electrophysiology side of things. This allows us to store cerebrospinal fluid, synovial fluid, blood, saliva, urine, fecal matter, muscle, skin tissue, lung tissue, epithelial tissue, tumor tissue. All of these various data types can be managed through the Loris environment. So at this point, you have over 35,000 specimens available publicly to anybody who signs up and registers, and you can get access to the actual tissue and biospecimens that are identified within the Loris database. If you want to find out more about this, the CBIG repository has been published recently in Neuroinformatics. Moving on beyond our own walls, the Canadian Open Neuroscience platform was launched back in 2018, and the CONP binds together a range of universities and higher education institutions across Canada to deal with four major domains: technical integration of data sharing and tool sharing, training, ethics and data governance, and then communications. So all of these components are part of the Canadian Open Neuroscience platform. I'll speak a little bit about this in more detail. This is the CONP portal where you can go and download any of these data sets. The bottom one here, for instance, Prevent AD, longitudinal multimodal study of pre-symptomatic Alzheimer's disease. This is rather different than either the CCNA that I described, the Canadian Neurodegeneration Network database, or ADNI, the US Alzheimer's disease database, in the sense that this is a database of subjects who are at risk for Alzheimer's disease because they have a family member who already has the disease. So this allows one to explore very early stages in Alzheimer's disease before it's become symptomatic. And obviously, if you can find out, you can identify its earliest underpinnings, then you have the chance to get in there and deal with it more preventively. Now, this dataset is publicly available. At this point, the CONP portal provides something approaching 75 different analytic tools. This is a change in clock ticking in months, so you see that there's a steady increase in the number of tools available, and in red the number of data sets available, which are up to about 50 data sets, and these increasingly growing every month now. On the training side, the CONP trains students in cross-disciplinary manners, so they can learn about imaging or behavior or genetics, so the geneticists can work with people in an imaging lab, and they can get cross-trained so that they become familiar and competent in multiple different domains rather than being siloed. And at this point we have trained over 63 different CONP scholars. We're also working, CONP is also working with the INCF, the International Neuroinformatics Coordinating Facility, to develop CONP curated training resources. So there's sharing of training materials between the INCF and CONP. And further work will organize, merge, and synthesize training resources to support best practices and develop skills for our early career scientists. A big issue in open science is the ethics and data governance of open science. Who has access to it? Who has access to what? And there's any number of issues related to data protection and privacy. The CONP has generated a number of documents on consent, on de-identification, and you can go to the CONP website to read these documents in greater detail. Much of the information in those documents is general in nature. It's not restricted to a Canadian context, of course. And lastly, within the CONP context, I can make mention of the open publishing environment. This is an environment that's called Neurolibre, and this is headed by Nicholas Stickoff, Rachel Harding, Pierre Belec, and you can go to the Neurolibre website and find out a lot more about Neurolibre as a preprint server for interactive data analysis. So this moves us beyond the idea of a publication being a fixed monolithic PDF submission to the journals. you can now work with the data and reanalyze the data with your own tools. It's the new way of doing science as opposed to a static publication. A number of journals have joined in with NeuroLibre to take advantage of its capabilities: eLife, PLOS, Magnetic Resonance in Medicine, Elsevier, and Aperture, which is the open science journal for the Organization for Human Brain Mapping. You can go to the CONP portal, CONP website, to check out these videos, which will give you a lot more information about the capabilities of the Canadian Open Neuroscience platform. The CONP is at root a platform for data sharing and tool sharing and training. It's general in nature. Any number of different networks can be built on top of CONP. They can be domain specific or they can be disease specific. One of the domain specific networks that has recently come into being is EEGnet. And EEGnet is obviously focused on EEG, allows it to share data of different kinds. And one of the best examples of this is the MNI intracerebral EEG atlas of Birgit Frauscher and her colleagues, a unique atlas from over 100 subjects looking at nearly 1,800 channels of intracerebral EEG. The overall goals of EEGnet is to provide a unified platform for sharing and analysis of standardized EEG datasets to the national and international research community. And it's has deliverables and milestones in the areas of data platform development, open EEG workflows that would run on CBrain, data sharing, which would be using Loris as the principal vehicle for data sharing, and again, analytics and standardized pipelines running on CBrain. And just to emphasize again, there's a lot of work going on in the world today to develop modeling and standardization of EEG practices and procedures, and EEGnet will be participating with international colleagues on this standardization move. For instance, moving and mapping EEG data into a standardized BIDS format has already happened, and we can expect a lot more of this standardization as we go forward. In the Chinese context, CBrain and our colleagues in China have been working with colleagues in Cuba as part of the so-called CCC, or China-Canada-Cuba network. This was inaugurated back in 2017. This is a brainchild largely of my colleague Pedro Valdez Sosa, who is working both he's Cuban origin, but also has a lab in Chengdu in China. And he and I have been working together also for some 20 years, so we joined forces to establish the CCC network, which allows us to take advantage of the tools I've been describing to process large amounts of data and share large amounts of data. Pedro heads the Latin American Brain Mapping Network, and you see here with a longtime colleague of mine, former colleague of mine, Keith Worsley, a wonderful statistician and mathematician. And what you see on the right here are the EEG sites across Cuba, all of which are networked together to provide EEG information in the clinical context for the entire population. The Cuban group have generated the Cuban Human Brain Mapping platform, and this is a deeply phenotyped sample of subjects who have behavioral, biospecimen, electrophysiology, and MRI information collected. And this data set was published recently as an open database in Scientific Data. Jorge Bosch-Bayar is a longtime colleague of Pedro's and also now working in my lab. And he's wrote the book on quantitative EEG analysis. And all of the tools that Bosch has developed with his colleagues on quantitative EEG analysis has been integrated into the C-brain environment for analysis of data resident in the loris environment. And you can see here some informatics pipelines and some of the databases which can be processed. Two of them right now, the Child Mind Institute's 1,300 data sets in healthy children, and the Prevent AD data set, which I've just been talking about earlier, on the at-risk for Alzheimer's disease elderly subjects. So all of the data is available in a BIDS setting within Loris and can be processed on C-brain using the EEG analytics that people like Bosch have developed. In the wider Chinese context, the CCC axis has nodes in the Beijing Normal University with people like Yang He and Galang Gong and Nishu and longtime colleagues and partners of ours, and they will be using this environment to integrate longitudinal EEG data in 30,000 children with collaboration from the CCC axis. Mu-Ming Pu, who's at this meeting, will be talking about, will be providing a mesoscopic connectome atlas of non-human primate data into the CCC and also what I will talk about in a moment, the Global Brain Consortium. On the wider international scene, then, these environments are being used to capture information from a variety of countries, subject them to analysis, standardization, modeling, and the hope, of course, here is to build up large agglomerated data sets which can answer questions which cannot be answered by smaller data sets by themselves. You have better statistical power. And of course, there are a variety of issues to be dealt with in terms of normalization and standardization. But this is the brainchild of Pedro Valdez Sosa and Min Li and Ying Wang. Going beyond the Canada-China-Cuba axis, there's also the Global Brain Consortium, which brings in colleagues in Australia, Canada, Europe, the US, which allows us to take the applications of EEG to a wider audience. The basic idea here is that the CCC axis is more focused on methods development. The Global Brain Consortium is more focused on the application of those methods in low and middle income countries, where often the technology, the most available technology is the inexpensive EEG technology rather than expensive MRI or PET technologies. And this is work that is being developed in collaboration with the World Health Organization. So, to conclude, I've talked to you about many different levels, the base level, the technologies that we use to allow us to handle and share large amounts of multimodal neuroscience data, as well as methods and projects and communities which sit on top of those infrastructures. And I hope you've found this interesting exposition of where we're at and where we're going. I will be forever indebted to all the people who do the real work in my own lab, as well as my international colleagues like Pedro Valdez Sosa. And it's been a pleasure to give this lecture. I look forward to your questions in the Q&A session to come up later. Thank you very much. Bye-bye.